So what were we talking about in 3E? What is this? This is 3E.
That is the way in which a virus reproduces inside a cell.
Totally. This is how a virus reproduces using a host cell, okay? This host cell depends on, the virus depends on a specific type of host cell. The viruses have receptors, docking proteins, that match the receptors on the cell. So that's why something like COVID. COVID is a respiratory virus. Why? Because the docking proteins, the receptors on COVID, match the receptors on respiratory cells. Hepatitis matches liver cells, or something like HIV matches white blood cells. So that's why viruses are tissue specific, is because of this docking right here. Okay, and since viruses don't have their own biochemistry, what viruses really do is they're molecular pirates is what they are. And the virus takes over the biochemistry of the cell. They take over DNA replication, transcription, and translation, except instead of the cell replicating its own DNA, and instead of the cell producing its own proteins, what the cell does is it produces viral DNA, viral proteins. These components are then assembled, and the virus then sheds or releases more virus. This process continues until ultimately the biochemistry of the cell is so disrupted that the cell dies. Okay, so this is how viruses reproduce. This is why also we don't consider them to be alive, because they aren't made of cells and they don't have the ability to reproduce on their own. So while we acknowledge the existence of viruses, we do not list them officially as a life form, even though when you look up microorganisms, viruses are listed in there as a life form, but they're not. Viruses are in this weird placement, okay? So this is any kind of cell, and this is any kind of virus, specifically a DNA virus. But there's two other virus types that we need to talk about. So this is image 19.5, and this is a type of virus that only infects bacteria, okay? So there are a group of viruses that only infect bacteria, and they are called phages. So this is figure 19.5, and this is your host. This is your host bacteria. Okay, so you and I, as eukaryotic organisms, don't have to worry about this virus because we are a eukaryote. Okay, whereas this virus only infects prokaryotes. This kind of virus only infects bacteria. And this kind of virus is called a phage. These are commonly called bacterial phages. Okay, but this is a type of virus that only infects bacteria. So a virus that only infects bacteria. Boom. And when you get the chance, you should do a Google search on these. Just type in phage, all right? Because they look like molecular spiders. We are not embellishing this. This is really how they look, okay? And then this here, this is the bacterial DNA hanging out in the cell. So the phage lands on the surface of the bacteria, okay? And if you notice here, it injects its viral genetic material. It injects the viral DNA, okay? This then codes for enzymes that digests the host's DNA. So that way the only DNA left in the cell belongs to whom?
The virus.
The virus, okay? So then you get the synthesis here. You get the synthesis of viral proteins and viral DNA. Good old transcription, translation, and replication. Okay. Once you have the components, you have viral assembly. And what happens is the cell literally fills up and fills up and fills up with virus until the cell literally bursts. It ruptures. So this causes cell death by lysis. And since the cell dies as the plasma membrane hemorrhages and splits, thus the Latin root for split, lysis, this is why we call it the lytic cycle. This is referred to as the lytic cycle because it is the viral reproductive cycle that ultimately ends with lysis of the cell. The cell splits because it fills, just like a balloon, it fills with virus and then literally pops. And then when this cell pops, it releases fully functional virus that simply bond or attach to other bacteria, and away we go. But that is how, so even, get a load of this, even bacteria have to deal with viral infections. Viruses, I mean, they attack everybody. They attack plants, animals, they even attack bacteria. It's amazing. But the whole, the general view here is that the virus, whether it infects prokaryote or eukaryote, the virus doesn't have its own biochemistry, okay? So what the virus does is it's a pirate, and it takes over the biochemistry of the host cell and uses that biochemistry of the host cell to simply manufacture more virus. And then the cell either sheds that virus until the cell dies, or the cell fills up and pops like an overinflated balloon. And that's why viruses destroy cells. Nasty. Okay? Any questions on that before we leave it? All righty. Do any of you recognize, let me zoom in on this. I don't know how much this is talked about in health class. Speaking of health class, thank you very much for your work with Mr. Miller, helping him out. It became very quickly obvious that he had the list that did not include your new credit. All right, does that virus look familiar to any of you? You've seen this classic image of this virus at all? All right, that's HIV. Okay, so this is now question 3F. We need to talk a ton about HIV, a ton of it, okay? So HIV has genetic material in it, and HIV is an RNA virus, okay? I think I should do that in black so that you can see it so it really jumps out. Boom, there we go, okay? It's an RNA virus, okay? So you know what's going to happen. This virus then is going to inject what into the host cell? What is that, right?
DNA.
Okay, DNA. It has to inject
Its own RNA.
RNA, okay? However, doesn't the cell need DNA to do transcription and translation? Yes. Okay,
Yes.
so that means that the cell has to go then from RNA backwards to what?
To DNA?
To DNA, okay? Is that usually the way it works? Does it usually go RNA to DNA?
No.
No, usually goes DNA to RNA, right? So therefore, the biochemistry that we're about to talk about here goes backwards. Instead of DNA to RNA, HIV is in a family of viruses that goes RNA to DNA. It does the biochemistry, what we consider, backwards, okay? Now, we have a term for going back. What do you guys call it when you wear clothes from like when I was in high school and you think that's funny? And wait till you're my age and it won't be so funny. What do you call when you wear old clothes, like from the 70s, 60s, 70s, and 80s?
Like vintage?
Yes. There's another word. It starts with an R. Vintage is a totally valid term. Retro. Yes. Okay. So HIV is in a group of viruses that are called retroviruses. So DNA is called, or excuse me, HIV is called a retrovirus. Why? Why do we call this family of viruses retroviruses? What do?
They have their RNA turned into DNA.
Correct. They convert RNA. They read RNA and use the RNA to produce DNA. They do the biochemistry backwards. So we then refer to all viruses that go RNA backwards to DNA. We call them retroviruses. Right? Now, there's some biochemistry here. There is this little receptor. Do you know how on the earlier image, where is that? There it is. On the earlier image, I said that all virus have these receptors on the outside, yes?
Yes.
Yes.
Okay. So that receptor there, Campbell's is showing you that receptor there. It's the same thing. Okay. So that receptor there is glycoprotein 120. One of the most heavily studied glycoproteins ever. It's called glycoprotein 120, although nobody calls it that. We simply call it GP 120. You learned about glycoproteins back in core 1 and 2, where the plasma membrane has proteins that stick out of the membrane, and then attached to those proteins can be sugars, and whenever you attach a sugar to anything, it's glyco, so that is a glycoprotein. Fine. So this is a receptor that is on the outside of all HIV virus. Okay. Now, that HIV receptor is specific to a receptor that's on the outside of this cell called CD4. The CD4 receptor is a receptor that's found on this kind of cell. What kind of cell does HIV infect?
Autoimmune.
Pardon?
It's an autoimmune disease, isn't it?
It is, which means it attacks your immune system. What do we call the cells that work in your immune system?
Macrophage or
Macrophages
phagocytic.
is one specific type. Go more broad. What are they all called?
Phagocytic.
That's another specific type. Step back and give us what's a more broad term
White
based
blood
on
cells.
how they look under a microscope.
White blood cells.
There we go. Well, they're white blood cells. So all white blood cells, not that macrophages and cytotoxic T cells aren't white blood cells. All white blood cells express this CD4 receptor. This is how white blood cells chemically talk to each other. Okay, this receptor is critical to allow your white blood cells to communicate. Okay, but guess what GP120 bonds to?
The receptor. Blood
To
cells.
that receptor. Okay, so how do you prevent HIV from infecting a white blood cell? The chemistry is extraordinarily simple. How do I prevent this virus from bonding to this cell? What do I do?
Block the receptor.
I block the receptor. If I block your CD4 receptor, that will prevent HIV from bonding to this cell, and you will not have HIV destroying your immune system. And they're like, come on, the science, this is easy. What's the big deal? Here's the big deal. The CD4 receptor is how all white blood cells communicate with each other. So if we block that receptor, we're shutting down your immune system. Okay, so the tricky part, the hard part about treating HIV infection is you have to inhibit the function of this receptor without shutting down the person's immune system. We haven't figured out how to do that yet. Because if you block that receptor, these cells can't talk to each other, and we just gave you medically induced immunodeficiency syndrome. We just shut down your immune system, and you're vulnerable to everything. Okay, and that would be bad. So if you could figure that out, if you could figure out how to block the interaction between GP120 and CD4, there's your treatment for HIV infection. It's a simple research question, but holy cow, I'll figure that out. All right, so I need a highlighter. There is an interesting addition to HIV. Can you see, and this is much more obvious in your textbook, can you see the little green dot right there, and little green dot right there? And then you can still kind of see if there's a green dot there and there, okay? Be sure, if you can't see it here, be sure to look at the image in your textbook, because that green dot is really important, all right? That green dot is also sitting right there. And that green dot is an enzyme, okay? Because the HIV lands on the cell, bonds to CD4. CD4 then literally opens the plasma membrane for HIV, and HIV injects its viral RNA, okay? Included with that viral RNA is this enzyme. This enzyme is a transcriptase. Do you remember when you did transcription and translation, and I asked you to define RNA polymerase as a transcriptase? And I said we would come back and talk about that later, and guess what? Now is later. So if this enzyme is a transcriptase, this is an enzyme that does what process?
Transcription.
Transcription.
Okay, except this is going from RNA to DNA. Does transcription usually go from RNA to DNA?
No.
No, this is going backwards, yes? So this enzyme is called a reverse transcriptase. It does transcription backwards, meaning that this enzyme that is contained inside of HIV is cheating, right? This enzyme copies the RNA and makes DNA. And so what you actually make here is this hybrid of RNA and DNA. Then the host cell replicates this DNA for the RNA, and you get here then viral DNA. And then HIV does something really, really, really sneaky. See how this is blue? This portion is blue. Do you know what HIV does?
Combine both.
Yes, it combines its DNA with the DNA of the host. So the viral DNA is inserted into the host genome. Do you think this causes the immediate death of the cell?
No.
No. Is that good news or bad news?
Bad.
That's bad news. Okay. So when we talk about Ebola. Ebola is an incredibly aggressive hemorrhagic virus. If you ever read about it, it's fascinating. It's horrific, but it's fascinating. It consumes connective tissue, and the person actually liquefies, right? So the bad news about Ebola is it liquefies you within about 2 to 3 days, okay? But the good news from a biology standpoint is it liquefies you in about 2 or 3 days. So transmission has to be very quick. So to treat Ebola, you isolate people for a very short period of time, and you wait for that virus to run its course, and then you're done. Your infection cycle is done. HIV inserts its DNA into the genome of the host cell, and then just sits here. And does this process look familiar? Viral genetic material, viral proteins, viral assembly, viral shedding. Does this look really familiar?
Yes.
Yes. Guess how long this process can last with HIV?
Are we talking about in just one single cell? How long it lasts in one single cell or in the entire body?
In the body.
Decades?
Yes. 7, 10, 15 years. Okay. So the good news about HIV is it kills the patient slowly. What's the bad news about HIV?
Kills the patient slowly.
Kills the patient slowly. Okay. And then, and you're saying, why is that bad news? Because that means that they're shedding virus. And that means if they are sharing their bodily fluids that have a high viral load that we'll talk about in just 1 second, then they can very easily spread this virus. Okay. So HIV literally hides, okay, it places its DNA into the genome of the host, and that allows viral shedding 5, 10, 15 years, depends on the age of the patient, the health of the patient, and the quality of medical care that they receive. Okay. So then here's, I mean, this is just good old viral messenger RNA, right? Which means that this is transcription here. That viral messenger RNA is then read to make proteins. So these are viral proteins. So this is then translation, good old transcription and translation. Over here, then you get viral assembly. And then down here, you have viral shedding. Or what is sometimes called budding. And this can happen for many, many, many years. And so the person then sheds the virus gradually. The HIV concentration in different bodily fluids gets very, very high, and they can then transmit the virus to other people. Okay. Now, there is some good news here, okay, in that the virus has a membrane. The good news about HIV is that it has a plasma membrane, which means it's sensitive to temperature changes and it's sensitive to UV light. Okay, so this little arrow, I'm going to draw it broader because this is a really big deal. The fact that HIV has a membrane is a big deal because that makes it fragile. Okay, have you heard the discussion about COVID, that when you expose COVID to UV light, what happens to COVID?
It dies.
Yes, it becomes inactive. Okay, what does that tell you about the outside of COVID? What does COVID have?
A plasma membrane.
Plasma membrane. Okay, whereas something like influenza doesn't have a plasma membrane. Influenza looks like that. Okay, so you blast influenza with ultraviolet light, no effect. You blast influenza with heat, nothing happens. Why? Because there's no plasma membrane. The influenza virus can sit on flat surfaces for decades, and you touch it, and you touch a mucous membrane, and you have influenza. The flu is a nasty little virus because it's so simple. All right, so how do you kill influenza? You liquefy it with bleach. All right, whereas the good news about COVID and especially HIV is, since they have a plasma membrane, they are not temperature tolerant. You start fluctuating the temperature, and that causes the membrane of HIV and COVID to break down. Now, this is really, really good news because mosquitoes, fleas, ticks, and any other bug that bites us and comes in contact with our blood, right? When someone who is HIV positive, if that flea or tick or mosquito takes in the HIV positive blood, okay, fortunately, the insect doesn't have a high body temperature. So when the HIV goes into the bug, there's a dramatic temperature change in what happens to the membrane of HIV. It breaks down. It dissolves, exactly right. Okay, and that's good news because embedded in the membrane is the docking protein GP120, which the virus needs the receptor to start the reproductive cycle. Okay, so when you talk about viruses, there's good news and bad news always. Okay, but HIV, what we call a retrovirus, it's a retrovirus because it does the biochemistry backwards. It goes RNA backwards to DNA. But notice what the theme is here. This is that whole molecular pirate that the virus can't reproduce on its own. So the virus has to take over the biochemistry of the host cell. The host cell for HIV is a white blood cell. Why? Because of the receptor interaction between GP120 and CD4. So that's the biology behind this. What we need to talk about on, we think when I'll see you next Thursday, is we need to talk about how HIV is transmitted. We need to talk about something called viral load. We need to talk about the fluids that you should stay away from. It is extraordinarily important that you guys are comfortable with how HIV is and is not transmitted, how you can protect yourself, because which age is the highest infection rate of HIV?
Teens.
Yep, yours. 16 to 24, by far. Okay, so unfortunately, you guys are in the group that is most commonly infected. So Thursday is going to be a little bit of a public health announcement because HIV isn't something you should be afraid of. You don't walk down the street and go, oh, I took a breath and I took an HIV and now I'm HIV positive. There are very specific patterns of behavior that will expose you to HIV. So we simply need to be sure that you are aware of those behaviors. And if you decide to participate in them, that's up to you. You're an adult. Totally up to you. Okay. So I think that's a really good place to stop. If you have questions on the extended essay forms, let me know. Please don't forget about the exam registration information. That is a simple form, should literally take you about a minute or two to fill out. Yet that form is super important for me, being able to register every one Saturday and Sunday.